Mix glycerol and dodecane-dioic acid in a 1:1 molar ratio at 120 degrees Celsius for 100 hours to obtain a polyglycerol dodecanoate polymer. Dissolve polyethylene oxide and gelatin in 65% ethanol with a weight ratio of 1.5 to 3 to 95.5 in a 15 milliliter tube. Tighten the cap and heat the mixture in an oven at 60 degrees Celsius for 1 hour, stirring every 15 minutes until the basal solution is homogeneous. For electrospinning, mix the PGD polymer and the basal solution in a 4 to 6 weight ratio. Add 0.1% riboflavin to the mixture and mix it well. Feed the polymeric solution into a 5 milliliter standard syringe with an 18-gauge blunted stainless steel needle. Then insert the syringe into a syringe pump. Attach the grounded lead of a high-voltage power source to the metal plate and the positively charged lead to the needle. Also adjust the distance between the needle and the aluminum foil strip to 15 centimeters. Now place the syringe pump at an angle of about 15 degrees with the horizontal to prevent aggregation of the fibers at the front of the strip. Turn on the syringe pump and adjust the flow rate of the pump to 0.6 milliliters per hour. Turn on the high voltage power source and set the operating voltage to 14.6 kilovolts. After the collection is complete, expose the fiber mat to UV light for 60 minutes of cross-linking. Transfer the fiber mat from the aluminum foil strip to a 100 mm Petri dish. Cut the fiber mat into round pieces of the same size with a surgical blade and place the pieces into a 24-well plate. Then expose the plate to UV light for another 20-minute sterilization. For cell pre-seeding treatment, immerse fiber samples in 1 milliliter of phosphate-buffered saline and incubate at 37 degrees Celsius overnight. On the following day, aspirate the PBS carefully. Add 1 milliliter of differentiation medium to each well and incubate at 37 degrees Celsius for 3 hours. After aspirating the differentiation medium carefully, add 0.2 milliliters of Matrigel to each fiber sample and incubate at 37 degrees Celsius for 30 minutes. Carefully remove the excess Matrigel from each well. Rinse once with 2 milliliters of differentiation medium. Add 1 milliliter of Accutase to the mouse embryonic stem cell culture dish. After 10 minutes at 37 degrees Celsius, when the MES cells are detached, add 4 milliliters of differentiation medium. Collect the floating MES cells into a 15 milliliter tube. Centrifuge at 400 Gs for 5 minutes. Resuspend the pelleted cells in 4 milliliters of differentiation medium.